So welcome to the afternoon session. And we have Dimitri Yatsenko who is going to present DataJoint workflows. So please take the floor.
Good afternoon. So it's a small group. There's a lot to talk about, and it's good you're here so I can see your facial expressions. Whatever, if I sense that it's not interesting, I'll move on to the next. Yeah, but please ask me questions if any specific topic, because we have a lot of different projects. If something particularly interests you, we'll dig deeper. So I'm Dimitri Yatsenko. I am the Chief Technology Officer at DataJoint. I've been working on it for a long time on a lot of different projects. We also have Tin here who will present more on the Tin Nguyen, who will present a little bit more examples live on the platform, how teams operate. And we'll really focus on the future, on the changes that are happening right now in terms of how science operates and how we can make it more efficient in every possible way. So these are the topics, and there are a couple of different areas. They're somewhat connected, but they're distinct topics. So if we want to spend a little bit more time on one or the other, the reason we combine these is because, well, we're working with a couple of teams here, and some contributed, for example, to the Microns project and are working with us with the Microns project. And we're also working on data archives, Dandy and Amber. So all of these are topics. But the main point is we're thinking of the future of research, and it is very clear that there will be new demands. Oh, and one more. Monty is here as well. Monty Cosma, he's also from DataJoint, and he does a lot of wonderful things, but one thing in particular, so we're here meeting with teams, is to establish partnerships and collaborations, especially in the space of research software. So we will spend some time today talking about MoSeek, for example. So MoSeek, and maybe I'll gauge the audience. How many people here are neuroscientists? Okay, most. Okay, so those who are not, are you more on data science, or I think?
Me?
Yeah.
My name is Susan. I was recently from the Women's Hospital. I'm doing AI medicine.
AI medicine.
It has a background at ICH. I'm here is really, I just found that topic is quite amazing. So to be here to think maybe something that we could use in the situation.
Okay, good. Then I'll explain a little bit more. When we talk about specific topics, I'll explain the basics, like what exactly we're talking about. And what about how many people study behavior, like video-based behavior? So how many people study? Okay. So behavior has been so about a third of you here. How many want to study behavior like video or plan to do it in the future? Kind of. I like that those who do study didn't raise their hands. Like they're doing it, but they don't want to do it. Okay, so no, behavior has been expanding modality in neuroscience because of first we understand, and I'll show you like the difference between natural behaviors versus the synthetic behaviors we've studied the whole time with buttons, and there is such a big chasm and separation from the difference between natural behaviors or naturalistic behaviors that you can now study with video is just so much richer. So I'll talk a little bit about how we address a lot of operational problems with using the DataJoint platform. And DataJoint Elements is a project that we conducted for about 5 years to create standardized solutions for data management in neurophysiology and behavior, and this is what's funding this meeting, and to disseminate technology, and in particular how to publish the data using in compliance with all the requirements. I'll talk about the Microns project. That's a somewhat separate topic but related, and it's a 10-year project that was just published in Nature last month as a special issue, and I'll describe as we get to it. And finally, I will talk about integration of AI into neuroscience from the point of view of managing the research process. AI is used in many different ways, but this is specifically in applying it to the process itself. So I'll tell a little bit about DataJoint. So DataJoint began as an open source project. I started working on it around 2009 as something for myself, but it's quickly spread to other labs, and it's a solution for managing data and computations together. It has a well-organized, systematic database and approach. Then we started this major project, Microns, that I was a postdoc at the time, that involved many labs. At the same time, we got funding from DARPA to start DataJoint as a company to support data-intensive projects. A couple of years ago, we got, or 5 years ago, we got this U24 grant to use DataJoint to implement standardized workflows. And then about 3 years ago, we got another grant to develop it into a platform that can be launched on the cloud or within labs. And so we launched the platform with several, so the open source solutions are in many labs, about 100 labs, so we'll touch a little bit on that work. But then we launched the platform on the cloud, the full platform, and brought a lot of our first batch of customers, first cohort of labs last year. And so in the focus there is really, so a couple of years ago or a year ago, most people were skeptical about the utility of AI as a real assistance in the discovery process, but now most people know that it's coming. So the mission of DataJoint is to create a robust operating environment for science, to make research more productive, more efficient, but also more rigorous, more robust, so that you don't, things like mistakes and data corruption should be avoided, and the entire process needs to be transparent and reproducible. So DataJoint itself is built on principles of some very solid computer science, the relational principles. I'll do a little demo to show how it works. And essentially, this is what we're calling a computational database. It's similar to how Excel works, where in Excel you have cells that have values and cells that have formulas. When you update a cell, the computations propagate and update. It's the same thing, but on the scale of big data, on the scale of big databases. So a computational process or a scientific process is expressed as a graph of nodes. Each node represents data structure and potentially some computation. So for example, green nodes represent a manual step like entering new data or acquiring a new file from an instrument. And the red nodes represent some computations that are performed on it. So, and then a lot of scientists now, a lot of labs have been using DataJoint to take their experiment and represent it as this computational schema. So if you are familiar with databases, this will be, a lot of things will be, and this is the next thing I'll ask. How many people here have used databases and understand like what a relational database is? Okay, about 5. Okay, well, our team I know does. So, so this is basically building databases where the data is stored, but also all the computations are tracked. And so you can build a computational schema that represents the logic, the structure of your experiment, and then you can publish the whole thing. You can share the whole thing, and you can aggregate the whole thing in your lab or in your life. This is how you accumulate your research assets. So I'll do a little demo. I'll go to the platform. Oh, I forgot to connect to the internet. How did I? I thought I was connected. I'll sign with a text message, or is it email better? Huh?
Yeah, you still need the email. Or text, yeah, that's right.
Either way. No, go back.
Sorry. I need to go back. I don't know how to go back. Oh, sorry, I didn't connect. I thought I was on. I've been here before. I should have something.
If you remember.
Should I just use my phone? Let me see if that works. Okay, I connected to something. All right, I hope my phone will be fast enough. So this is what computational database. This is, for example, this is the MoSeq pipeline. We'll discuss about MoSeq a bit today, but this is a complex pipeline. Each node here represents each of these oval shapes represents a module. So it's a Python module with a collection of classes that represent tables in the database. And also, so you can go to the code that created it, and also it stores your data. And so again, all of this can reside on the cloud, as it is the case here, or you can set this up in your lab. But this represents the flow of your pipeline. So I will use another example. So the cool thing that is, I work with a lot of projects, and I can switch between them, and I can immediately understand what is going on in that lab, in that project. I don't work on as many projects as Tin here, because Tin, his job is to work with all the labs and to help them operate. So he probably has the record for the most projects he's working on. But I'll show a very simple one, building from scratch. I'll just build an entire pipeline from scratch. So right now, let me using Python code. So I'll go to the notebook, and this particular computational pipeline is very simple. It creates and manages different Julia sets. Do you know, who knows here what a Julia set is? One? Mandelbrot set? Okay, yeah, so Julia set, yeah, okay. So Julia, there's a programming language called Julia, but this is, a Julia set is a mathematical concept. Let me,
Maybe if you switch to internet,
Hmm?
maybe it's slow. Refresh the page.
Okay. Tin, look into it. I'll move on and let me know what the okay. I can go to Notebook Plus. Yeah, please look into it, and I'll come back to it. So I'll show you a couple of other pipelines, just example. Oh, no, now it's coming up. So this is Jupyter. How many people here code in Python? Okay. How many people use Jupyter? Okay. So, all right. Then I'll explain a little bit. So Jupyter has been one of the most transformational concepts. It's a concept called literate programming, invented in the 70s. But it's a combination of storytelling, graphics, and code in one place. So I will use that example to create a complete of. All right. So first I'll explain what a Julia set is. I'll make it a bit bigger so you can see it. All right. So a Julia set. So right now I initiated some libraries. Okay, I'll let it load. Sometimes the first time when it takes a while. So Julia set is a very simple formula. So I'll explain right here. So this is the. I cannot leave the microphone. Yeah. Okay, so this is a very simple formula that produces very beautiful images. So fundamentally, you start with a kernel. Here it's C. It's a complex number. And then this X and Y generates a mesh, X and Y coordinates, a mesh grid, basically an image. And then you combine, you apply this recursive formula. It's right here. And then you go through the cycle, and in this very, very simple loop, it generates images. But right now, the notebook is not loading it.
You just have to restart the notebook server.
I have to go to launcher and restart it.
So you can go to File, top left. Yes. Go to Help Control Panel. And then just delete it. Yeah, that one.
Stop.
Yeah, stop it. Restart.
Okay, so that will probably take 2 minutes. Okay, then and then restart.
You can delete that as well.
Okay.
Now you close and open
Start new server.
just close that tab. Close the entire Jupyter Hub tab.
Jupyter Hub
The
tab.
browser tab, sorry.
Okay, okay,
Yeah, close this browser tab as well.
okay.
And
And just go back to Works.
yes.
Okay, so I'm going back to Julia said. Yeah, okay, let's go to Notebook.
And let it restart.
Okay. All right. So this allows you to, with everything, well, typically it works right away. So we have this notebook that runs all the time, so it comes up right away. And then Notebook Plus, this is for complex computations that require GPUs and things like that. That one is a bit even more finicky, but because you have to start an expensive instance, so it usually takes like 2 minutes to start up, so you can do your analysis, and it automatically quits, so it's not always up like Notebook is supposed to be up the whole time.
Just binary, you can
Oh yeah, it's because I'm connecting through my phone, and let me come back to it in a sec. In the meantime, I will show a couple of other projects just to illustrate. So also on the Works platform, but I'll show some live projects that are working.
This part is not part of the paid solution, and this is free to use. Will you show us that?
I will explain what is included soon. It's coming up. So there is everything that touches your data, and all the code that the pipelines, everything is under your control. That is what the DataJoint platform creates, are all the tools to orchestrate it, to manage it, to share it, all of those types of things. So I'll explain exactly how that works. And so let me show a couple of projects, like real projects. So SRI, Shirley Ryan Ability Lab is a clinic at Northwestern University. There's James Cotton is Dr. Cotton, so he runs a clinic where they collect videos of people who are recovering from stroke or from injury or from surgery, and they need to learn to walk again, and so they recover their gait. So in this case, he created this pipeline, and that he uses this in his clinical practice, and you can see that there are green steps, that's where the data is acquired, like videos, for example, and red steps, that's where computation is performed. And the idea is that when new data is acquired in these green steps, so sometimes a green step may follow a red step, for example, when you need to validate a bounding box around a person, that's a manual step. So this represents the logic of his experiment, also the data in it, and all the computation that's associated with it. And so you run this pipeline, and then, but he created this because he's a good programmer and he knows DataJoint really well. But the people who use the pipeline, they use the dashboard. And the dashboard is used to, maybe I should connect to the,
Yeah, I'm connected.
maybe I should take some time to connect. Sign with email.
Your text message.
First, we need to agree to the terms of service.
Oh, yeah, that's it.
Sign with text and then
Text, and
You
then
still have to put in your
the mobile number is below.
you still have to put in your.
I see.
I should get any text or something.
And now you all know my phone number, so you're welcome to to call.
We blur that when we publish the tape.
Oh, okay.
I already had it.
All right. Okay.
No, it should have been a success screen.
Yeah.
Maybe you need to try that.
I haven't
No,
had.
no, not this one.
Sorry to make you watch this.
Exactly.
Okay.
None of the calls now and stuff.
Yeah.
It's good to see the news struggles.
Yeah, we all struggle. It's a struggle. Okay, there's the success screen. All right. Okay. All right. So the way researchers interact with it, when they interact, and in many cases, as James himself, when he meets with patients, all he needs to do is enter the new session, and then the rig records videos. The videos are automatically uploaded, and the pipeline automatically triggers and percolates through all the steps of the analysis. And then you build custom custom dashboards. So Plotly Dash. How many people here have used Plotly Dash? Okay, one person. It's like two. So Plotly Dash is similar to Matplotlib, for example. So it's a library for visualizations, but that's very cloud friendly, or web friendly. It's basically for creating web applications, dashboards, and visualizations. So the data comes from the rig. So in this case, this is not a patient. This is a graduate student. So you get the raw data, and then you get the output of the analysis, which is the same person with all the body tracked. And so this is really transforming how we're working with rehabilitation data. Instead of bringing people and gluing fiducial markers on their body in a special room, you can now do the same thing with just iPhone cameras and then you get these models that can then be further quantified, like their gait can be quantified, so you quantify how long the person, how much time the person spends on each foot, like the pressure distribution, all those types of things. And then you can track recovery in the effects of treatment. So this is one example of how a DataJoint pipeline can be used. So let me switch back to our Julia dataset notebook. All right, so I'll go to our notebook, and we'll build an entire pipeline from scratch. It's very simple. Notebooks. All right, let's see if this will execute now. Okay, it executed. All right, so this is the function that creates a Julia dataset, and I will call this function with this plotting function, and this is what a Julia set looks like. Can you believe that this one formula right here, or actually this is the initialization, this formula right here that's iterated in a loop, for some magic reason, it creates these beautiful, beautiful images. And if you change anything in this, so one thing is these images are infinitely detailed. You can zoom in forever, and there will be continued detail. And if you change the seed, you'll get very different patterns. So with a different seed and a different zoom factor, so this is zoomed in 10,000 times, you get a very different parameter. So here are a few examples of different seeds, and I iterate through them, and depending on the seed, you get a very different parameter. So now let's build a DataJoint pipeline managing this. And so I will, so this is what you do. The first thing, you import DataJoint, then you declare a schema. A schema is basically one node in your pipeline, and we'll only have one node with a few tables. And the first thing I'll actually do, I'll drop it, so we'll start from scratch. So schema drop means you have nothing in the database now. and then I create it again. So if I go to my pipeline visualization, there is nothing there. So, and if I go back to the notebook, and I should keep the 2 tabs open the whole time. Let me make that as a separate tab. So it doesn't have to reinitialize. Okay. Oh yeah, I know what I did. It was in the other one. So notebook. So, and this should be executed. All right. I'm here. So I created my schema. And here I create the first table. So in a table, you declare the table structure. That's the definition. So a table is a table in the database, but it's a class in Python. And then it's a manual table, so which means I enter data into it manually, and I can insert several of them. And then, so this is, if I go back to my pipeline now and refresh, I'll have my first, this is my schema, and the first node, this is the Julia specification. So this is my, then I define the second node. The second node now points, this is the definition, it points to the table above and adds 2 new fields, the image and a PNG, which is a saved image, compressed version of the image as an attachment. And so, and computer tables define this make method that has 3 parts: get the data upstream, perform the computation, and save the data. And so I just defined this, node. So if I go back to my pipeline, now I have 2 nodes, and I can look at the data structure. So right now I'm in the structure layer, so if I open a table, I see its declaration. So if you're familiar with relational databases, this will look very familiar. Or I can go into the data layer. So my Julia spec, I have 7 specifications for Julia data sets. But my Julia image data right now, image table is empty. So you can set up the pipeline that computation will get triggered immediately after new data appears upstream. But here we're performing this step manually by saying populate. So what populate does for a table, a computer table, it calls make for all the data upstream that is available. So and it will display the progress. So there are 7 images to populate, and it exits it with 6x count of 7 and no errors. And so if I look at my Julia image table now, I see that it populated the values. I can fetch the data now and visualize it. I can go back to my notebook, see those. Oh yeah, because this is science, we added some noise to this image. Because in science, you don't get perfect images. In math, you do. So this is a Julia spec image, Julia image with some noise. The problem with Julia and fractal data sets is there's no characteristic scale or frequency, so it's very difficult to separate noise from detail. So it's very difficult to design a noise filter that would remove the noise but keep the detail in a fractal. But we will try. So there are different, we create now a lookup table. So lookup tables is another type of table that has fundamental facts that are not part of your data, part of the data definition. So this defines several methods: a Gaussian filter, a median filter, a wavelet filter, and a total variation filter. So, and then I design a 4th table now called Julia denoised that points to the Julia image and to the denoise method, and now computes the denoised image. And here I have my make function. And the make function gets the image, gets the denoising method, and depending on the denoising method, performs the computation and saves the result. In this case, the saving of the result is a little bit more involved because it computes, because it plots the image and saves the figure. And this is the last step, which inserts the data into the pipeline. And so I have 4 denoising methods, and I have 7 images. So how many denoised images should I have? 28. So it's all the full combinations. If I look at my, so I'll run the populate. And so now it's for every denoise method and for every image, noisy image, it performs all the calculations. And then if I look at my Julia denoise table, it has 28 entries. And now I can get a key, which is basically the identifying information randomly. So here's the first one. This is like Gaussian filter applied to a noisy image. Here's another one, and so forth. Basically, I can, for all the 28 of them, I can look at all of them. But because we also have our, so let's go back to the pipeline. This is our full pipeline in this case. So it's Julia specification, then compute a table that computes the Julia image with noise. Then we have a denoise method. So the direction is always bottom or top to bottom, and you build a database that recomputes the data anytime new data appears. Do you have any questions about now? Do you have a new concept of computational database?
Right. So basically you populate this image programmatically. Does it work both ways? You could first draw something and create a programmatic
No. So no, we don't have like, so it's, right now this is just for navigation, but you create everything with code.
Okay.
And this is for actual collaboration. So it's always, so every project consists of a Git repository and a database. And that's matching is, so you can, for every project you go to the code, and the code is a GitHub repository, either private or public, but so there's full integration with GitHub in this case. So a lot of processes are, so when you set up a project with DataJoint, it's a GitHub repository, a database on-prem or in the cloud, but there needs to be that matching. So this is just a simple example, but let's see what scientists have done with this now. All right, now I'll move quicker. So the main idea is that, well, let me talk about the infrastructure. So I think I'll answer a lot of your questions, Peter. So fundamentally, a computational database consists of these elements. So first, there is for the first time, we're developing to a full spec. So we just published it this month. So if you go to DataJoint specs, this, if you're a computer scientist, this has the full definition of what it means to create a DataJoint project. So it's an open standard that you can use to create computational databases. It's separate from the platform. The platform uses the spec to operate it. And then DataJoint Python is the reference implementation. So right now, it's the spec is a bit ahead of the implementation. So the spec contains a lot of the new features that are coming up. And there's a discussion group there as well. So that if you have some inputs on the new features or new capabilities, you can discuss them there. But fundamentally, the project needs to have these components. It needs to have a code repository where the team manages the code that defines the structure of the pipeline. It needs to have a relational database that manages all the dependencies. And the database structure is constructed from the code. So the database runs by itself. You don't need to do much with it. It's automatically managed by the code. You need to have an object store. This is where the big data, so we call this object augmented schemas. This is basically relational databases are not really designed to handle movies and electrophysiology data. So it's a combination of object store. An object store can be a file system, but more modern object stores like S3 or Ceph, they have additional features. But it could be basically a network attached storage system. And then computational dependencies are embedded, but it's an important concept. It's really not a separate construct here, but I want to highlight it here. So computational dependencies are effectively inferred from the schema in the database. So this process captures the code, the data, and the dependencies. So this type of approach now, the DataJoint approach of this type is in about 100 neuroscience labs. Actually, we don't keep track of all of them, so maybe more. Lots of papers have been published, so it's been around for quite a few years. So now let's talk about the next layer. You can now build services around it because this core is really designed for research engineers. They build, but to make it useful, you need graphical user interfaces, you need these cool things, you need ELNs, electronic lab notebooks. So you can add an electronic lab notebook. It's an interface that creates or that helps you enter the data, visualize the data. That's what you just saw in that dashboard. So our dashboard is our electronic lab notebook. So if I go back to the, am I still running that simulation? So if I go so the dashboard for this guy, is the Julia image. So I can see the results of the analysis in graphical form. So scientists don't have to go to Jupyter and code in Python. The research engineer configures the dashboard, and they can see the results of the analysis. So, and we'll show beautiful examples of how many teams have built for their projects. So students run experiments, but the PI can go and see the results of last night's analysis. Yeah.
Can you maybe, if you can show the, back to the dashboard, one on the other page?
Okay. Dashboard. Dashboard, yeah.
So the data entry tab for it?
Okay, so you can enter a new seed, like a new spec, and then you can, once you submit, you can rerun the analysis, and then you will see the new result. So right now it's not automated, right? No, okay, but it could, so in many cases, or how we set this up, when you enter new data, the computations kick in automatically. You don't need to go to the notebook and types populate. So we basically keep track of new data, and it calls the code automatically. And that's how a lot of labs operate.
And what we use here is using Plotly and Plotly Dasher. These are open source tools. You can build, the intention is for you to build your own dashboard here, right? So and there's components that are DataJoint aware. So that form that you see is just based on a table. So Dimitri defined the table behind the scenes. And then that page with all the entry forms is just based on that table. You don't have to write much additional code to put together that page. That's kind of the point. So in DataJoint, you just write the table definition, your data models once, and then you can easily create dashboard on top of that. You can create visualization and workflow orchestration. That's kind of what we're trying to get at.
That's right. So on the outer ring here, what you see here is not standardized. So the inner core is very standardized, and that's what touches your data, that's what manages your data, that's yours. But what people have done over time, they build a lot of tools around these pipelines. And before we had our platform, many labs already were using it. They built all sorts of things like the staging space, that's effectively, that's where the raw data is before it's structured. Job orchestration tools, Slurm, all types of things. Publishing interfaces or visualization interfaces, and integration with the instruments. But now with our platform, we've built our way. It's part of the platform, so it is proprietary to DataJoint. But the data and the code that runs the pipeline, you can still run it by. So if you run the platform, you can still run everything manually. But with the platform, you just get a lot of these cool new features. So these are kind of functional extensions to the core that make it easier for a lab to operate. And finally, on top of that, what has emerged over the last couple of, a lot of progress has been made over the last 6 months or a year. How effective these AI tools have become at co-piloting and basically understanding your data structure, and your code at the same time. And so, but integrating that on top of your project, so these co-pilots become fully grounded in your project, makes them immensely powerful. So we're working on making that a native part of the platform, so you can interact with but there are many, many steps that go into this. So how many people have worked here with coding co-pilots? Okay, good experience? Mostly. So they help you become more productive, and they definitely will not replace you, but they can make you a lot more productive. And so then labs use the DataJoint platforms to build their operations in the lab, using basically around the platform all the interfaces to the instruments, to the electronic lab notebooks. That becomes the system of record for your lab, for yourself. So this manages your scientific or research assets, your code, your data, your methods. And then you can host it in the cloud, you can move it to the cloud, you can move it between institutions, and those types of things. So fundamentally, it is the researchers that create the pipelines and manage them, and this is their baby. So this is from next door. This was a couple of years ago, Bernardo Sabatini's lab, who launched their pipeline for a lab of about 50 people. And implemented in there to run their experiments. So now we'll talk, so I described the framework and the platform, but now these are the specific things. So we've worked with a lot of labs who created their beautiful pipelines, but they were not standardized. So 5 years ago, we got a grant. It ends today with this meeting. So after 5 years, we ran this project to create all these pipelines. And we call it DataJoint elements, and this is for all the, well, the reason that the project itself ends, but we made it sustainable. That was one of the purposes of this project. So this is to design modular pipelines that would be community-driven. So we have a scientific steering board who provides constant input for us in covering the basic modalities that are involved in running a typical systems neuroscience experiment. This usually involves a neurophysiology modality combined, well, sometimes with anatomical, but more and more with behavior. So these are standardized modules that use uniform processes so that you can publish data the same way, enter the data the same way, independent of what kind of experiments you run. And then because they're modular, you can design pipelines, like for example, this one, that combines many modalities. Behavior, operant behavior with buttons and pushes, fiber optometry, for example, for stimulation, calcium imaging, electrophysiology, so that's in the same experiment. It doesn't all have to be in every mouse in every session, but this pipeline is capable of supporting experiments that combine for these. So a lot of labs are using this. These pipelines rely on a lot of open source tools. And we develop joint sustainability roadmaps with labs. So when we work with, say, spike sorting, then we have a relationship with a spike interface, kilosort developers, tools that are used for to implement this. So this is from a couple of years ago, but there are many, many more tools, but all of these tools are used. So let's look at specifically at MoSeq, because Barbara is here. Barbara is running at Harvard, is running the behavior core. Hi, Barbara. So we spoke yesterday about deploying MoSeq as a service by the behavior core. So what that could be, so what MoSeq is, you have, and this is specifically key point MoSeq, you have a video of a mouse with no markers of any kind. You can have a key point tracking machine learning algorithm. Deep lab cut is one of the most popular, but there's also sleep. How many people have used deep lab cut or sleep? 3, 4, yeah. So this keeps track of all points. And then, but what MoSeq does downstream, so it was developed by Bob Data's lab here at Harvard. So it's an unsupervised machine learning algorithm that segments continuous behavior, because the positions of the joint they're not meaningful. But it's an unsupervised learning algorithm that breaks down the behavior into short episodes called syllables, things that you can then interpret. You can fine-tune the algorithm, and so maybe Tim will show how that's done in the pipeline. And this is on the left. You see is their documentation. So we work closely with the data lab with Bob Data and his students and postdocs. And this is the DataJoint pipeline that reflects those steps. So Mackenzie Mattis, who developed Deep Lab Cut, she's been using DataJoint since at least 2017 and 2018. And so a lot of her experiments that she was running to develop Deep Lab Cut were orchestrated in DataJoint. So this is the part where this is the training pipeline, and this is the inference pipeline. So there is a training step where you teach the algorithm to separate behavior into episodes, and then the inference when you have new behavior and you want to separate it into episodes. All right, so one important development, kind of jumping around. So something that we found that many, many labs use years of their lifetimes is when they don't apply standard quality control metrics to their data, and they collect, say, electrophysiology data for 9 months, and then they realize that their ground was not done correctly, and they, or something else, and because they don't have these metrics all the time, so the same, and I'm one of those people. I lost quite a bit of data in my PhD when there was a warp mirror in the microscope, and for a year I was collecting data that where the point spread function was so long in the axial direction that it was measuring signal above and below the cell just as much as inside the cell. And so I had a whole theory developed from that data, which was destroyed by replacing the mirror. And so we developed, for the principle is for every modality, you need to have stringent best practices for quality control, so you detect these problems right away. It's probably one of the most biggest reasons for inefficiency in science. Underappreciate, yeah. Because people, when they find something like this, they're embarrassed. They hide it and just move on. But for example, for 2-photon microscopy, we introduced this metric that all the signals need to be translated into photon counts. Using Poissonian statistics, you can infer the magnitude of each photon. So we published this with a bunch of other co-authors in Nature protocols. And so now, if you see anyone publishing their data, and if it doesn't use real photons as your bar, uses like AU, arbitrary units, then you should ask them to redo this. Because what when you run the analysis like this, so the sensitivity of the microscope depends, a 2-photon microscope depends like as the 4th power of the numerical aperture, as exponentially decreasing with depth. And if you don't detect these problems, so this is part of the microns data. Here, these big events are on the order of 10,000 photons per second from each cell. And that's one of the best. And you can have 2 labs using the same type of microscope with the same lens, with the same objective, but when they're not doing something right, you can have an order of, or 2 orders of magnitude difference in the quality, and they would not notice because they look at averaged images and the data is not resolved. So this was from 2023, so this event is sponsored in part by the Open Data in Neurophysiology. We had another event in 2025, but Nima, where's the paper from 2025?
We just released the revision.
Updated?
Just like 2 days ago.
Okay, I didn't see it. So ODIN is an initiative that run here, is centered here, but this is the effort to unify the ecosystem or to inform the ecosystem of tools. So this is a preprint, you can find it, but you said there is a 2025.
It's supposed to be published
In Neuron, okay. So this is an ecosystem of tools that interact, that are interoperable, and we very much operate in that same space. Another big initiative is data publishing. So how many people here have heard of Dandy? A few, so now you know. So Dandy is an NIH-funded archive for publishing neurophysiology data. There's a new one called Amber. It got funded about a year ago. It uses the same architecture now, and I think as of recently. So Dandy is run by the main PI is Satra Ghosh, who's partially here, partially at Harvard, correct? And then Amber is the principal investigator there is at the Applied Physics Lab, Westerbrock. All right, so that's that, and we will be meeting with them to. So here's the thing: a lot of labs are not using these formats. Sometimes they're not required. Sometimes they're not required to submit. For example, Amber is primarily supporting this Brain Initiative project called BBQS, Quantified Behavior Quantification Project. But a lot of big projects. So there's a new project starting in Europe, but a lot of labs are here in the U.S. Called SCENE. It's 20 labs working together. It's funded by the Simons Foundation. It's getting funded this month for 10 years to use machine learning to infer foundational models of behavior. Mackenzie Mattis is the principal investigator or the lead investigator, but the total, so you can look it up from the Simons Foundation. And they're not required to publish data in this format, and so they will be submitting it to Zenodo and to other things. So So we want to work with, this is the plan, with Amber to make it easier across different, and basically to make, to unify different ecosystems. All right. Any questions about this? Next I'll talk about the Microns project briefly, and then we will, how many people have read, heard, learned about the Microns project? Okay, then about half of you, so I will start from the beginning. So the project started, the discussion of the project started very early, 2013, 2014, and then it got funded in 2016. And the idea is this, that, well, Francis Crick, back in the 80s, wrote a book, and there was a line there in that book. You know, that said something to the effect that, you know, the ideal experiment would be to record from every neuron, say, in a cubic millimeter of cortical tissue and map all the connections, then we would really learn something about the brain. But of course, it's impossible, so why bother? And now, 30 years later, or 40 years later, we had the technology to do it. And the combination of calcium imaging and electron microscopy provided that ability. So the goal of the experiment, of this study, it was funded by IARPA, Intelligence Advanced Project Agency, to the tune of about $100 million to between 20 universities initially, to record the activity of every neuron in the visual cortex, from a cubic millimeter, every neuron. It's about 120,000 neurons. And then do detailed electron microscopy. And I don't know if my claim is quite correct, but close to that. This is probably the most studied tissue in history on that scale. So the data, this came out as a dedicated special issue of Nature on the 9th of April, 2025, a couple of months ago. So this has been called by different, so it was in Reuters, New York Times, and everywhere, but they call this Apollo Project of the Brain. And so the lead institutions that were doing this, so I was at BCM, and DataJoint was used for was the system of record for all the neurophysiology data, calcium imaging. Then the Allen Institute ran the neuroanatomy, and then Princeton, Sebastian Sung's lab, did the machine learning and the inference of all the meshes and the morphology and the connectivity. So this is the data that was published, and this is how you can access it. And I'll explain a little bit about how to work on the data, and a little bit about the project that we are running currently with NEMA together with Neuromatch to analyze some of these data. I think these data are very, very rich, and new papers will be published for the next, probably with an exponential decay, but with a time constant of, well, maybe it'll be an alpha function. Big burst followed by exponential decay. But I don't think it's used to the extent. Actually, the most papers that have been published have come from from studying the vessels, because it also uncovered all the vasculature in the brain that had never been studied at this scale. So there are 9 different data types. So the functional data in response to the visual stimuli, electron microscopy, then the match between the functional data and the electron microscopy, then the cell segmentation data, then the inferred cell meshes from that, then the synapse connectivity from that, then the identification of the nuclei, and then the proofreading that is still ongoing. And then cell types. Actually, a couple of new cell types have been described based on this data set. So there is a new project, ongoing project called Vortex, which is the Visual Observatory Cortex, run by the Allen Institute. So what they do is, when you have a scientific question about this data set, you submit a request, and they do the morphology proofreading, because the machine learning was not perfect. The original data was not perfect. So they have a whole bunch of proofreaders, I think in Indonesia, if nothing changed. and they keep track of, and they visually verify that all of that the inference of the shape is correct. And they have a review process, so it's a funded Brain Initiative project to do that. And then we are hosting a data set, and we are working with a copy of that data set to work with. Oh, do you want to take a picture? Sorry. Alex. Yeah, this is also, this is in the main paper. I don't have the citation here, but this is the lead paper in that special issue of Nature. It's on the cover.
And that paper, by the way, the link to that paper is in the Micron Explorer link.
Yeah, so if you go to micronsexplorer.org, you'll have pages and pages of information about all of this. So I'll just briefly summarize. So these are the visual stimuli that were shown to the mouse. There are two types of novel synthetic stimuli. I designed them specifically for the purpose of quick inference of classical receptive field properties orientation tuning types of something we've studied for the past 60 years in mice. And then also videos, and there are several types of videos, synthetic videos like video games and movies, and then natural scenes. So we had like Mad Max, Matrix, you know, a couple of movies that we split into 10-second episodes. And that's what and so a lot of studies were done with that. So this is how to access the physiology data, not the morphology, not the connectivity, but the physiology data. So the main way they published, because it was done in DataJoint, DataJoint is a relational database. So the main method how they published, they just dumped the database into a SQL file, and here you have it. So for you to reconstruct that you need to set up your own SQL server, load the data, and then use the DataJoint API to access and work with the data. So that's difficult. Method number 2, we host a copy of the data we already preloaded it. So, and you can access it with it's read-only, but you don't have to set up your own server. So we set up a dev container code spaces. How many people have worked with code spaces? 1, 2, okay. So code spaces, dev containers are a way to have the entire environment for your coding preset somewhere, and what GitHub allows you without anything else to just code right there. So if you follow the instructions in this link, you'll be able to just walk through a tutorial how to query the data. Easiest. So then a few very nice people converted the data into NWB files and uploaded them into Dandy Archive. So there you can download it in NWB files, and then you have your own copy. You don't have to work with the database. And then there is a fourth way, Pro, and that's what I'll talk. This is working with a complete data pipeline. So the thing is that even though when we publish Micron's data, it's one mouse, but hundreds of mice were imaged, and only one went all the way through. So that's the $10 million mouse. But we have a lot of, $1 million mice and and $100,000 mice that have excellent data. They just didn't go all the way through. And for you to get a lot more statistics, you need to use more data. So that data is not published as part of this data set, but it follows the same format. And so we've set up a way to collaboration between Neuromatch and Stanford, where Andreas Tullius is, where the data was first acquired. Now he's at Stanford. He was at BCM. And this is the project we're running. So there's Monet and Trippy stimuli. They're not analyzed that much as part of the main paper because everybody switched to analyzing live movies. But immediately we discovered something we did not expect. It could be sad, but it's also interesting. These stimuli were designed to, the alternative, they're basically both decorrelated noise. Their power spectrum is roughly the same. It's decorrelated white noise with different correlation structures. So one is just basically moving optical flow, and the other, there's like local contours. And these are pixel-wise responses, as you can see. So blue here means responding. These are pixels that are sensitive to natural movies. And most cells, you probably don't see it as well as I can see on my screen. Pretty much most cells respond reliably to natural movies. So we measure reliability by playing the same movie twice and seeing if the traces are correlated. And this is basically what we're plotting here is pixel-wise correlation coefficient. And so, and you see that, so blue is response to natural movies. Green is a response to the trippy movies. And red is the response to Monet. So I don't know what you see here. Do you see how many blue cells versus green cells versus red cells? Most of them are blue, so that means that cells respond to natural movies and ignore the other types of stimuli. And for 60 years, we studied gratings. I spent 4 years collecting data, and gratings are roughly like, and so for a long time, we studied responses to gratings. The problem is that the brain doesn't care about gratings. And so, but what was surprising, do you see, what color are you not seeing on this, in this? If you will see this, there is no yellow. So yellow is a combination of green and red. So that means that if a cell is responsive to the Monet stimulus, it does not like responding to the TRPS stimulus. This is not predicted by any theory. It actually contradicts. If cells are filters, they should respond to everything. These are very similar movies in terms of their spectral content. They're identical. They just have very different structure in global properties. So, and we found this, and this is sad because from one point that all the history of the past 60 years of recordings, we were learning from just a very small number of cells. But the interesting thing that these two stimuli, not by design, by a little bit of serendipity, seem to uncover two different subcircuits in the brain. And so this is what we're studying here. So if you plot the responses to Monet on the x-axis and to TRPE on the y-axis, there is nothing on the diagonal. So basically cells, so what we think we're into, so this is unpublished data, this is going to our SFN, this is our SFN poster for this year. And so this was done we're working with Paul Fahey and Talia Mohammed, who collected the data a few years ago. And then Moore and Rana and Mohammed from through Neuromatch, and Nima and Andreas. And so here and now we want to interpret this, understand where this comes from. And so one hypothesis that could explain this is that potentially things that are cells that respond to Monet are cells that respond to just optical flow, like when the mouse moves its head, everything is moving. Whereas the green responses, the TRPE responses, correspond to detail, something that objects in the field. And we actually don't know how to interpret this yet. So this is an ongoing experiment, and there's a ton of experiments that you can do with this. But this data is available basically as part of the microns data set. So understanding now the unanswered question: are these cells different morphologically or in terms of their connectivity? So if I look at all these red cells, I can now call the Vortex program and say, here are the cell numbers, please confirm, give me the morphology, and you can query them now. What do these cells look like? Are they different? So that's how you can work on these pipelines. So I can show this pipeline in the platform. So if I go back and go to the TRPE Microns project, this is the pipeline that produced this analysis. So if I go to the pipeline, so this is the experiment data. This is the pre-processing data. Here we used compression and quality control to be able to do pixel-wise analysis and segmentation. So this is the pixel-wise analysis that is performed. You can go and see the code. So right now we didn't make it public yet because we hope to publish this as a paper, but we will release this. So right now it's a private repository. But you're welcome to join and study this deeper and maybe do additional experiments to figure out what these two functional subcircuits are, because this is very surprising. All right, I don't want to take too much time from Tin, but so I will keep going.
Go for the AI.
Yeah, so I want to talk about AI for sure. So this is, Neuro AI is next. So we are going through a major shakeup in science. I don't think there's a single cause. Not one person is causing this. I think there is a fundamental shift in how science is done. The effectiveness of science, the old model is really, we need to reinvent things. And we've been thinking deeply about this. We got together a couple of years ago with several collaborators to define what PsyOps means, basically a very efficient way of doing science correctly on scale. So we published this preprint. It's been sitting in Nature Methods for about a year now, but we hope it will come through. But the main thing that we define the progression of labs as labs. Most labs are at this initial level of organization. Most labs right now have very bespoke, basically ad hoc development and processes. They, over time, mature labs, established lab become more managed. They have more repeatable processes, so they don't start from scratch every time they start a grant. Then they go to a defined level where they use standards, fair data, fair workflows. And then they go to processes that are scalable, where you can capture entire workflows. That's just what we call PsyOps. It really starts at level 4 and 5. And level 5, now a lot of labs are finally approaching it. This is where that scalability also is combined with tools that complete the discovery loop, where AI is used tools are used not to just analyze the data, but also to direct the focus of research, to steer experiments. So this is called closed loop science, closed loop discovery. So as a caricature of the scientific process, you can think of science as a loop of deductive reasoning, which, if you start like good science should be hypothesis driven. That's what we learn in school. But where does the hypothesis come from? It comes from insights that we learn from the data, and until now, only humans could do that. So that right part, we have textbooks on statistical methods, how to go from a hypothesis, how to design an experiment, and test your hypothesis from data. So that's the right part of the loop. The left part of the loop, when you look at data and see something and go, oh my God, what is that? That has never been formalized until now. So basically, this is what AI methods do for us. They help you just by looking at the data, detecting anomalies, and formalizing them into phenomena, and then perform inductive reasoning. So inductive means to go from the specific, like seeing something, to general, like a hypothesis. Deductive means starting with the general hypothesis and designing a specific test that should fail if that hypothesis is not true. And so this complete cycle, now you can design basically your systems to complete that cycle. So this is what we call like the 5th level of PsyOps, and it really depends on machine learning and AI enablement to complete the process. So specific examples is a series of experiments that, again, my mentor Andreas Tolias is running, again, using DataJoint. These are closed-loop experiments where the stimuli are determined by the analysis of the neural activity, and there are many such new closed-loop experiments. And finally, this is where I want to have a bit more. This is one of the final points. So how integration of AI, right now there's a lot of investment into AI co-scientists. And this is where we'll distinguish 3 levels of how AI is applied to manage the scientific project. At the first level, level 0, we'll talk about AI Copilot. This is a tool, AI tool that can help you manage to basically, this is where rigor and precision is very important. This helps you code your pipeline, understand your data, work with your data, perform queries, and analysis. This is your Copilot when you code that helps you write good code. The second level, this is where a lot of industry investment is going on right now. So projects like futurehouse.org, for example, NovelSeek, which is a Chinese AI system that was published just last month. Ready Set Potato, this is, you might know, Nick Edwards. He was a neuroscientist at UCSD, and he started a company that just got funded called Potato. The website is Ready Set Potato. I don't know if it's a pun on Apple but it's potato. And the idea is to have closed loop science. Google co-scientists, if you Google that term, you will see a whole bunch of good reading from DeepMind and also from Google AI on managing the scientific process, and then the Microsoft Discovery Platform. So these are just some examples, but these are all industry-funded, big, multi-billion, probably in valuation, probably in the $1 trillion range. So this is nothing compared to the academic projects that are funded. So these are, and we probably will not compete with them. We will integrate them. So this is one cool thing about how AI is evolving now. It's AI systems are becoming, they're competing with each other, and they're becoming more commodity. And the teams that will win, they will be teams that will use these tools effectively. And this is good. This is good for science. We just need to build processes in our lab. Right now, you can probably afford to ignore it for another few months. But in the very near future, if you're not using good AI tools to help you with your science, you will start getting behind. And this is becoming clear. A year ago, it was not clear. Now it's kind of, all right, it's probably going to happen that way.
We don't know if it's good for science.
I don't think there will be an alternative, basically. So to do good science, you will need to. And then we'll talk about the third higher level. This is more of a strategy level, and this has to do. So we go from AI co-pilot and AI co-scientist to AI co-visionary or co-strategist. This is to understand if what you're doing is significant and and what you should be doing and with whom you should partner and what grants you should apply for and if you should be developing something that the industry needs, what patents can you uncover. And actually, a bit more progress is made on that. So we are partnering with this company called DeepInvent, and I'll demonstrate quickly how that will look in the future. Actually, it looks very good now. So DeepInvent, one of the co-founders, Marcus Weller. So I just ran a couple of. So for example, what this allows you to do is to start with an idea. And this is, again, if it's fully integrated, this could be an idea coming out of your research. And then it analyzes this idea, maps the space of current IP. And it learns more about the company. And the company here, it could be your lab or your institution, to understand your capabilities, how you fit with others. Does it make sense for you to file for a patent, or does it make sense for you to apply for another grant? Then you go through the process of studies. I'll skip the company step here, but in this case it's DataJoint. So reports, it learns about DataJoint's capabilities. But you can put your lab there, and it says, okay, you are well positioned to do this. So, and then it does the background analysis on existing grants, publications. So their technology can be directly applied to do grant applications, or is basically the same graph, knowledge graph analysis in projecting in time how things related, where the field is moving. And let's generate, it's already generated. Prior art, so existing patents, so that to see if you have something so the next step is to innovate, to find what has not been. And I actually don't want to show this because this is work from this is, when it generates, it says, yeah, this is patentable. So I will probably not scroll down all the way down. Then, and again, I haven't worked through it, but you can edit this. At each step, you can edit. And then the next step will take your edits into account. And then, so alignment is that alignment between the need, the capability, and so it analyzes that space and then aligns your capability, it recommends what you should apply for. And the next thing, it does writes a provisional grant application. We will generate one. So this will generate knowledge that no one has seen before. And let's take a look. Should take some time, so it's telling you what it's doing. But the idea of integration is not to do it from scratch from a few ideas, but to ground. So if you have a DataJoint project, you have your co-pilot to help you code. Okay, so this produces a pathway for me how to create dynamic neuroadaptive pipeline integration for in neurorehabilitation in this case, for example. And this basically gives me a summary of what I need to be doing to apply for a patent and whether it's worth it. And then the next step will be to write a provisional application. And the next step will be to generate the application to file to the US PTO. So, but the main point is that by itself, this is very powerful, and this, again, this platform launched last month, and then it is gaining substantial traction. But the same can be done in the scientific domain as well. The power of this will come by integrating it stacking it on top of your co-pilot and your co-scientist that keep track of all the findings. Because how many patents do you apply for 1,000 experiments? Probably 0.1. And so it takes a lot of ideas before you generate something from which a commercial enterprise can be launched. So, but we all now know that we'll be doing science differently because funding is becoming a lot less open in terms of what you do as a scientist, and labs need to become a lot more versatile, switching between basic science, applied science, industry-funded research, SBIRs, basically launching companies, filing patents, and licensing their software. So it's generating the provisional application. I mean, it may take 20 minutes, but it would take me longer to actually, if I had to write it. So here it is. And then the actual application. So yeah, so this is the process. And then
if you have a patent idea, you go backward.
So every time you go back, if you go backward, it generates a new branch. Yeah, so it will keep the old branch unchanged. But if you make modifications, if you go back, it will generate a new one. So yeah, I think once we have integrated, so right now most of the work where we are focusing is on the co-pilot level, just helping people work with their data and make sense of it effectively, because the data will become more complex. All right, so areas for collaboration. So the way we work at DataJoint, we are researchers, but we're also a commercial company and developers and collaborators. We apply for a lot of grants, mostly as co-applicants. Co-applicants providing the engineering and the data infrastructure. Have you heard of the gold standard science?
Yeah, the recent executive order?
Yeah. So this is an executive order that was issued a couple of weeks ago from the White House. And the idea is that they're giving the NIH and the NSF and other agencies 60 days, I think, to issue new directives. So within the next couple of weeks, we will have new directives about, there are 9 principles there, that science needs to be transparent, reproducible, that uncertainty should be quantifiable and made obvious, that conflicts of interest need to be obvious, all of those types of things. The NIH right now, in terms of AI, has been very conservative. They have a specific prohibition against using AI in grant evaluation or grant writing. But they know they need to get out with the time. So right now they have this, request for information that's due on July 15th, asking you, everybody here, what are the principles for using AI for science? What safeguards should we have? So that means that in the next months, we will have a lot of new direction coming, how science should be done, especially how AI should be integrated into our processes. But I think it's coming in a very strong way. So one of the requirements there in all of these is operational excellence. If you receive funding, you'll be expected to have solid data governance, the infrastructure and the operations. So this is what we help establish, that governance and operations. And then implementing this AI-powered closed-loop science. There are lots of grant opportunities currently, both from government funding, but also from lots of foundations. So one thing that is changing is philanthropic foundations have now matched. They have been rising in relative and absolute terms, to catch up to the amount of federal funding, but also a lot of industry partnerships. So industry-sponsored research, if you're using good processes in your lab, then it's much easier for you to get business from a pharma company or to perform research for them, basic research or applied research. Then in the areas of research communication and publishing, so AI will transform this. So writing papers, once you can explain a new finding to AI, then they will be able, then AI systems can now, you know how you use, I don't know how you use Gemini or ChatGPT, you can take the same ideas and now present them in 5 different, now do it in 500 words, explain it to a 5-year-old or now, draw a diagram. You can, as long as you can capture your job now as a scientist is to spend less time writing papers, but much more time solidifying this knowledge graph, communicating it to a system that can then present it in a variety of ways. And if we now are training systems to be both to present graphical information, generating new ways of graphically representing the data, generating figures. So I think that research communication process and reviews, and the same to the reviewers. You know how communication now happens? If you ask to write a 50-page report, you write an outline and ask your ChatGPT to generate a 50-page report, and then the reader will take that 50-page report and ask their ChatGPT to convert it to bullet points. And the same, so the review process will also look in very much the same way. As long as we will learn to use these tokens, basically tokenizing our findings in a way that we can track them, that will become the foundation for publishing much faster peer review. And so that's coming. We don't know what it will look like, but this unit of a PDF published in Nature as your accomplishment, that takes a very long time, will likely to be transformed. That's a very old and antiquated system to disseminate communication, research communications. And social media is not the right answer either for communicating your scientific findings. You need a system that does have verification, peer review, but is much more flexible and responsive. And technology dissemination. So we develop a lot of technologies, and there's many ways to disseminate them. Something that scientists are not very good with is how to do it well and how to generate revenue for their labs. So something we're here to, we have been doing, for example, with Harvard is pioneering this as basically the software that you're generating. Peter, we have a lot of questions for you about that. You can add an agreement to your license. It's free for non-commercial use, but for commercial use, here's how this is where you pay. This is how you use it. So I think more and more institutions, definitely Harvard needs it right now, will be, and so MOSIC is following this model right now. So Monte here worked with the Technology Transfer Office, is that what it's called? OTD, to develop this model where MOSIC is free for all of you to use, but if a commercial company wants to use it, they can work with DataJoint, and then the revenue is shared with Harvard and with their lab as well, and so, but it's free to all non-profit research institutions. So the same model, I think all scientists need to become more informed and capable of when they generate new solutions, they need to put those clauses in because your future research may depend on it. Funding will become more nuanced, let's just say that. All right, so this is just some of the things, but this space is quickly evolving, and science will be different. Science will be more important than ever, but scientists will need to become, we're developing this vision more of scientists as entrepreneurs, scientists who manage their assets, their findings, their data, their code, their workflows. That's the most precious thing that you have in your career. And as you come out of a training program as a postdoc or graduate student, it's not just your CV and publications, but also all the data, all the workflows that you know how to execute, that you need to learn how to aggregate that and communicate it. So this is what we're helping, and we're figuring out, we're not just helping, we're working very actively with all of the researchers to figure out how that will be done. Thanks so much. I took a little longer, but Tin will actually demonstrate a bunch of projects, like how they use this process. Let's take a break.
Any questions or anything?
Yeah, any question? Oh yeah, there's supposed to be a Q&A. Any questions? Yeah.
Can you talk a little bit about like how that integrates with, in the lab, people always have their own like workflows, tools, things that, codes that they've worked on for years, or this kind of stuff. And that's usually a big barrier in adopting new methods, new tools, because you think like, oh, why should I, spend so much time adapting and learning new things because I already have something that works. So
Yeah.
how does this like,
I mean, if your system corresponds to all the principles of gold standard science, then you should keep it. We hope in most people, in 99% of the case, it's not. It's not something that can be. And so developing that process that can be done that way takes effort. And so we provide that uniform approach that they can. It's uniform in the processes, but very adaptable to any science that you're doing in methods and integration. So yeah, it takes some time to re-engineer. So Monty uses this term re-engineering. What is the term?
Use that term. It's fine.
Re-engineering SciOps, re-engineering scientific operations. So re-engineering is something that labs will have to do to meet a lot of these standards and to become more capable.
I mean, like one good example is Marshall's lab at Johns Hopkins, the same Schuler lab, where we started working with them about June last year, and it took us about 60 days to get all their pipelines built and running. And then by December, Marshall couldn't believe it. Like, he was looking at the volume of data across and said this would have taken us years. They had this big backlog of data, and then they were able to go much farther and faster than they expected. So that's the part of the acceleration, and that's even without the AI analysis that now they're doing.
Yeah. Yeah.
And can I ask a question? You mentioned AI Copilot, that's 3 layers.
Yeah. So co-pilot is the one layer, the first layer, then co-scientist, and then co-visioner and co-strategist. Which one is better? Yeah, one of those. Yeah.
So the question is, so the scientists involved in DataJoint will have access to these APIs. That's what you mean.
Mm-hmm.
Okay.
Yeah, so this is, right now, it's a collection of tools that are not yet fully integrated. So this is where we actively, I think in the next few months, this has been our priority too. So right now we have tools that are tools like Cursor AI that do very well with the code, but they don't do very well with the data and the data structure. And so to work well with the data, you need another translation layer. So these are, for example, one paper we just published with our collaborator, James Cotton, who was mentioned here. He developed, so you need to be able to convert your data into tokens. And so this is done by these transformers so that are specific to your data that you need to train. And so what some of the grants we're applying for with partners is to develop a standard process for training these translators. So he called his BioMech GPT, and the paper just came out as a preprint in May. And so BioMech GPT helps you translate gait and various movements, biomechanical movements, into English or into tokens that can be done into any language. And so that you can ask questions of the pipeline, show me all the episode where the person was limping or was on the left side or something like that. And it can translate that into queries to the database. Your standard Cursor AI or all the other current pilots cannot do that. So there's a lot more, and for all scientific data, you have to do that. So this is where we're not producing the complete solution, we're producing a roadmap, how you can, because you own this is your baby. So what scientists here at MIT, at Harvard, everywhere will be doing now a lot more is training AI to understand their data before they can move on to the next step. And then with that data, with that translation layer, then these types of tools can be more effective. And then once these tools translate into findings, then translating that into significance. So what it means. Why should you do this? What grants can you apply for? What medicine can you do? What drugs can you develop? That's more on the level of strategy.
What's the difference between this and agent?
Agent, so all of these are agentic AI. Yeah, but now, so agent is a more general term. But so all of these are AI agents that are grounded in a context. So the good thing is that the field has converged on a few standard approaches how to do it. This MCP, this model context protocol type of thing. So there's more so these tools have become more pluggable. But the main limitation is data, how to structure the data, how to present it to the AI. And because the science is complex, much more complex than a lot of other things, you need this. The problem is that the constraints here, this can be more creative. This needs to be precise. You cannot afford hallucinations here, but here hallucinations are welcome. You're exploring. Well, not welcome, but they can generate new ideas. Here you need many external resources that are ongoing. You need to know what grant solicitations are open, whether you're a good fit for them, and so that's on that level. So I think this is actually, we have better solutions for this than for this. And this is absolutely fundamental, but we kind of know how to do this. We just need to put this together into a complete system. So this will work. It's hard, but it can be standardized.
We need fundamental models to make sense of the object store.
Yeah. Yeah. So like AIs are like current ones that can make sense of figures and kind of from a figure explain. But if you want to go from the raw data to interpret it, yeah, so this is what a lot of models are getting.
I mean, I think it's going to get there at some point. Because,
Maybe.
I mean, I cannot imagine everybody have to train their own little thing to interpret.
Well, just for a specific domain, once you train for that domain, once you train it by a McGPT, you can train it on a whole bunch of different human, like, yeah, it's generalizable, but general models don't do that. Yeah. Like surgery, planning, and, So, yeah, I think lots of models we'll be developing models for the rest of our lives. That may be a long, long time. Maybe, if we solve aging, that means we'll be using them forever. All right. Anything else? Just need to solve the funding problem, and this is why you've noticed that there's a lot of focus on funding, how to make scientists more entrepreneurial. Huh?
Why don't you use these files? I mean, any reason that you only show these files?
Which ones would you?
Yeah, these five here, so actually have some.
These are examples, but what's your favorite example?
Just so you understand that. So what's your criteria? I mean, which is these five?
These are the ones I read about. I haven't read a whole lot about others. Does anyone else have a favorite co-scientist model?
Yeah.
Here, Nima is developing a new one. What is it called? I'll add it to the
It's
list.
called HIPSIN, so it's hypothesis synthesis.
Hipsin.
It's part of this MIT.
Okay, so I will go ahead and add it right now.
You have a website or you have
So we, and because we apply for grants and our role, we apply for grants because that's how we develop collaborations. HIP,
That's correct. S capital.
SIM,
Sin.
Synth.
Yep. That's it.
So we have a new one called HipSynth that I'm yet to read about. So does anybody else have a favorite?
Undermine, it can help you research a topic. Undermine.
Undermine? Yeah, so I think deep research does a pretty good job, and there's a lot of ChatGPT models that can do literature review. But by these I mean more of a something that manages your findings and helps you generate new designs of experiment, something a little bit more. If you read this paper, Novel Seek, it's pretty fundamental about the problem with a lot of these solutions. Why they're I think Microsoft fundamentally will try to drive cloud usage. They run everything on their cloud. So we don't quite like that. We want to be able to use different clouds, not be locked in. and enable scientists to run on their infrastructure.
Where do you place notebook LM in there?
Yeah, so Notebook LM, again, I don't think it implements, it can be a tool that can be grounded in you. Like, there may be a way using APIs to use it in this context, but there's a lot more work. So I think for Dimitri, a lot of these are LLMs that are just grounded in the right context. So it's a lot more, yeah. So it could be used for it, but not by itself, not without a lot of work to embed it into your process. All right, let's take a break.
Let's take